From Microbe TV, this is TWiEVO, This Week in Evolution, episode 120, recorded on November 26, 2025. I'm Vincent Racaniello. You're listening to the podcast Behind the Biology of What Makes Us Tick. Joining me today from Salt Lake City, Nels LD.
Hey, Vincent. Great to be with you. TWiEVO 120, man. We are
It's a good number,
getting
right?
long in the tooth. I love this. Speaking of podcasts and numbers, did I see that Immune is about to celebrate its 100th episode?
Yes. Yes, indeed. We are doing 100. We're going to have a
Wow.
live episode at the incubator in December 22nd. Yeah, everybody's going to
Whoa,
come. We're going to have two local guests. Should be fun.
beautiful. I'll be tuning in for that one.
We
And I have to
have,
say, oh, yeah, go ahead.
we could put about 25 seats in the incubator. So we're going to rent some chairs because I don't have 25 chairs.
Very cool. I'll be, yeah, kind of, some fun energy. Fill up the incubator. I won't be there in vivo, but mark me down as an in silico
In
observer.
silico.
Yeah, yeah.
Silico.
I almost said in vitro, but that wouldn't make any sense. But yeah,
What's,
and I have
what's
to
your,
say,
what,
Vincent, I
what's, no, sorry, go ahead. Go ahead.
just have to, I love the sort of holiday adjacent podcast. So here we are, on the edge of Thanksgiving, and there's something in the years past, we've done some episodes of TWiEVO right up to the holidays. I kind of have in my memory some of our first, discussions about SARS-2 right around the holidays as that was coming on
Yeah.
the radar screen. But anyway, the energy at campus kind of winds down. Half the people are on the road, on the way to catch up with family. I think it's a perfect time to have a fresh podcast sort of arrive as you're traveling, or as sort of you turn down the volume on the rest of work life, regularly scheduled work, etc. And so anyway, just good to be together.
Yeah,
Yeah.
I'd say it is. Here in the U.S., we celebrate Thanksgiving tomorrow, the
Mhm.
Thursday, and I don't mind having a podcast the day before. We're going to do TWiV on Friday. I wouldn't
Cool.
even mind doing one on Thanksgiving, but nobody would listen. But hey, you record it and it gets released. Because why is
Yeah.
that? You may ask. Because it's my greatest joy
Yeah.
is to do these podcasts. So why not tap into your great joy, right? That's what life, you only go around once in life. So you have to
Mm.
grab everything you can from it,
Mm.
right? Nels, 15 degrees
Um,
C and cloudy. I'm home today because
yeah.
the trains are too crazy. It's full of people with big
Oh, yeah.
suitcases trying to get places. Oh my gosh, this guy last night in front of me had
Mm.
the biggest suitcase I've ever seen in my
Mm.
life. Like, what are you going away for a couple of days? You really need all that? Oh my gosh. Anyway, it's too crazy. So I just said,
Yeah.
screw it. Do my two live streams here.
Good call. Hopefully there's some, among those travelers, there's some earbuds in that are tuning in to Microbe TV podcast, this one included. Here, the weather, we're waiting for winter. So kind of on the edge of, we got in the forecast some storms on the horizon, but the models are kind of confused, and we're waiting to see if it comes in as snow, as it comes in with rain, but a little overcast and right around freezing here in Salt Lake at the moment.
So let's see tell us where you're from, folks.
Yeah.
We have a fewer people than usual, but we still have people. And
Yeah.
let me thank our moderators here today. Our moderators are including Les.
Oh,
Thank you
great.
so much for joining us. Tom is in Eugene, Oregon. So Tom, I'm
Fantastic.
going to Eugene
Hmm.
next May. We just made the person in the department just made the travel arrangements
Great.
for me.
Hmm.
So let's see if maybe we could, if you're in there, we'll connect and maybe we could hook up. That would be fun. Here's the link for today's paper. By the way, this is a fun paper. You'll like
Yeah.
it. Barb Mack is here, UK. Barb Mack, UK. Thank you so much
Awesome.
for being here.
Wintry England.
And let me show you. I'm going to show you something that Barb Mack had
Ah.
made, because this is good, because not only is Barb Mack here, but also the person who drew it, the artwork I'm going to show you,
Hmm.
is here.
Fantastic.
So Barb Mack is right there,
Yep.
and you remember, you may not remember, but a while ago she drew a picture of Amy Rosenfeld, the Black Essay Princess. Okay,
Wow.
she
I
had
love it.
someone draw
Yeah.
it.
Yeah.
It was
Yeah.
drawn
Yeah.
in an anime style. Okay,
Sweet. Hmm.
and now she has had this individual Uba Luby, who's in Little Rock, Arkansas.
Oh, wow.
And
Welcome.
she drew what I'm going to show you now. Thank you so much,
Hmm.
Uba Luby. Let me get up the picture here. Where is it? Office. I put it in my office hours folder. And it is kind of like a summary of my career in a way. Where is it? Can I find it? I thought I put it in here. Right, it's 2025. Oh, 11/26. That's
There you
here
go.
we go. Vincent Art. I'm going to just pop it in here, and I think this will work. Here we go. There you go.
Oh, wow. Look at this.
I
Yeah.
have to shrink it, don't I? Here we go. We can shrink it, and then we're going to see Nels and me.
Oh, wow.
Maybe we'll make it big so we can see it.
Yep.
So
Move
this
around.
is
There we go.
drawn
Mhm.
by Uba Luby. Oh my gosh! Thank you so much. I love it.
Fantastic. Oh, my
And we
goodness.
have a lot of so this is based on Cells at Work. I really like that manga
Huh.
series.
Hmm.
And so you can see here we have different characters. We have a macrophage here. I
I
like
love
that
it.
they're
Yeah.
ghost-like. There are a couple
Oof.
of macrophages.
Yeah. In the
And
battle.
here is a neutrophil on the way to eliminate a filthy germ, because the germ here is taking over the nutrients, right?
Yeah. Hand over the nutrients. Wow.
And here we have this chipper erythrocyte is delivering oxygen to a delightfully passionate science podcaster.
Oof.
TWiEVO, and we got TWiEVO here. See, how about that?
Fantastic.
And I
Love it.
wonder who that would be,
Mm.
but that's very cool. And then here,
Hmm.
platelets, the body's vital construction crew. Despite being quite small, the body's cells look up to them. Platelets love their big sibling, red blood cells.
Wow.
And
Fantastic.
then here is poliovirus down at the bottom. We have several poliovirions. The causative agent of poliomyelitis. This picornavirus encapsulates its plus single-stranded RNA genome with an icosahedral protein coat and enters hosts via binding the poliovirus receptor CD155.
Mm.
So there's a shout out to me and my lab. Of course, we
Wow.
identified many years ago the poliovirus receptor. So thank you, Barb, and thank you, Uba Luby, for this. This is a lovely holiday gift.
Incredible.
I really appreciate that. I will show it again tonight
Wow.
in our office hours. Yeah. Isn't that cool?
That's spectacular. Yeah. That's
But,
spectacular.
have you
And
ever seen the Cells at Work manga, Nels?
I actually haven't. I'll go look that up.
Yeah.
And Vincent, but put up Ubalubi's comment there, because there are some, from that fantastic image artwork. There's also some ties to our episode today. So we're gonna have some platelets in the game. We're not gonna have a polio virus, but we're gonna
here
have a
we go.
foamy virus.
This one. Good
Yeah.
afternoon
So
from Little Rock. It's a bright, sunny 11C. I wonder if the rainbow fish from that children's book had a retrovirus-driven trait variation.
there you go. There's some echoes of the viruses are everywhere, as we've talked about for years now, and we've got a good one today. Should we jump right in, Vincent, or do we still have a few more?
Let's see if
Yeah.
we have a few more people here. Yeah, we've got Uba Luby in Little Rock, which I
Great.
visited a couple of years ago. I did the
Mm.
Marie Chow Memorial Lecture
Very
a
cool.
number of years ago.
Mhm.
Pete is here in London,
Great.
where it's 6C and dark. Oh, yes, this is funny. Les noticed today we released Twin 66, the neurobiology of overeating. Perfect
Oh, there we
timing for Thanksgiving, right?
Another holiday adjacent podcast. These are some of the best ones.
I'm releasing like one a day this week, because it's just the timing
Yeah.
of them all.
That's great.
Kent, the cat guy. Thank you for your
All right.
contribution to
Wonderful.
science communication. We appreciate it. We need your support to do these sciencey podcasts, and really grateful for it.
Fantastic. Yep.
Claire is in the UK.
Welcome,
John
Claire.
is in Des Moines, Iowa today.
All
Oh
right.
boy.
Welcome back.
Kent the cat guy is from Bellingham, Washington. 8C and rainy.
There you
Right. Barb
go.
Mac didn't draw
Thanks, Kent.
the Amy. You had an artist draw the Plaque Assay
Yep.
Princess. That was very good.
I see.
That was very
Yeah.
good. Yeah, it's Eugene. I just was arranging the
Hmm.
flights. So they had wanted me to fly to Seattle and then drive 3 hours to Eugene. And I said, no, I could fly to San Francisco and then fly to Eugene. I don't mind
Yep.
taking a stopover. You know about that, right, Nels?
I do. That's a good call. And actually, I'm just trying to track down my calendar. I'm going to be in Eugene sometime, I think, late winter, early spring as well. Hosted by a former postdoc from my lab, Matt Barber. It'll be out for a seminar. So I'm predictably unorganized here, disorganized, and I'll get those details and TWiEVO's ahead to put that in case we could potentially connect in vivo. That would be a lot of fun.
Yeah, that'd be cool.
Yep.
Ubalubi, thrilled you like it. I'm so happy to have the opportunity to make it. Well, I'm really appreciative of you doing it. It's a lot of work, obviously, you could see.
Yeah.
And so thank you so much. It's great.
Great detail in there.
And Hatamama is in Western Massachusetts.
Welcome.
Welcome. Did you see Vinny's glucose parlor sign?
Mm-hmm. Yeah, as you were scrolling through that image, about halfway down in the middle
Really?
is a sign for Vinny's. Yep, go up. A little higher. There it is. Now zoom in right in the middle, that purple sign. Vinny's.
Oh, there it is, right there. Vinny's. Oh, wow, I missed that. I guess there's a lot of things I missed here.
Yeah.
So, yes, Uba Luby says, I've hidden quite a few Easter eggs in the art. One of my favorites is a vertical farm in the top left corner as a shout out to our favorite Dixon. Oh, there you go. There's a vertical farm. That's so cool.
That's fantastic.
Love it.
Wow. Yep.
So everything means something here. That's great. I'll have to take a closer look at it. Yeah. Nels spotted it. Yes. This is so good. It is a really great piece of art. Thank you so much. Love it.
Yeah, agree. All right. Well, we've started to assemble here. There's good energy from all around, all points on the map, and let's get going. Building off of Luby's great artwork, we've got some platelets, Vincent. We've got some viruses, and we've got some fish. Another great topic. So, as you know, we're always on the prowl for interesting evolutionary genetics, evolutionary biology here at TWiEVO. I had the fun opportunity to connect recently with the senior author on this paper. This is Molly Schumer. She runs her lab at Stanford University. She's incredibly gifted, multifaceted, multidisciplinary evolutionary biologist, a lot of field work, working with especially swordtail fish, species and strains of swordtails that you find in rivers and tributaries in Mexico, Latin America. And this work goes all the way from the field, collecting individuals from various populations, down to the molecular detail of what is it that contributes to some of the really charismatic traits of these fish. And so the title starts to give it away here. Insertion of an invading retrovirus regulates a novel color trait in swordtail fish. This is one of those, to me, one of those preprints or papers that is perfect for a holiday episode. It has that sort of energy or feeling of unwrapping a scientific gift, of just learning something new, seeing how these surprising connections under the hood of some of the traits, the color of fish or the pigmentation of fish that we're really familiar with. And, I think Tom was hinting at it, the iridescence, this fascinating, the shimmer or the sparkle that you can see associated with the scales of some fish is just really, these charismatic traits that are really inspiring. And of course, as we're familiar, the viruses have something to say about this, as they do with a lot of evolutionary biology. How do traits sort of arrive or get? What are the genetic changes that impact that? And so we'll return to some of those ideas. But before we get there, Vincent, let's talk about who did the work. You can see the list of the authors here. So it's a international cast of scientific characters from different points on the map. Um, some co-first authors, co-corresponding authors. In addition to Stanford, we have researchers who are from the Nanjing Normal University in China, Texas State University, the Chinese Academy of Fishery Sciences, the Benamarita University near Veracruz in southern Mexico, the Institute of Fisheries in Puebla, Mexico. And so, I want to pause here for a minute to talk about Major League Science. This is how in the US, historically, traditionally, up until today, we've practiced science, is with people from all over the world coming together, contributing in different ways. I think I missed it. There's also researchers from Germany who included some, the resources to do some of the electron microscopy here. And so, this is it. This is how, like, part of the magic of science that's been unfolding for decades in the US is that sort of welcomes all comers. And as we've been talking about on TWiEVO, it feels like that sort of style of science is changing right before our eyes. The attacks or the bridges that are being broken between researchers in the US, in particular researchers in China. A lot of skill, talent, resources, incredible opportunities are sort of, potentially sort of falling through the cracks here, or worse, the roadblocks are being put up to make it harder to embrace these kind of studies that we're seeing here.
Not only that, Nels,
Yeah,
but you could work on whatever you want, including these fish, right?
indeed.
A swordtail fish, and nobody would. Well, some people would criticize you, right? But I have a feeling that this kind of work is going to be even harder to sustain in the current climate.
Indeed. And so let's talk about the funding. So here at the core of this work is federal government funding from the US, NIH, NSF, Germany, private foundations, in philanthropy. So let's unpack that for a minute here. So NSF, National Science Foundation, GRFP awards. These are fellowships to graduate students. We just had this year's competition. It was chaos. It was a mess. So they changed the rules in the middle. They were really unclear about whether you'd submit here or there. All this new information right at the last second had to be put in certain formats. So we've taken, I think the competitiveness of science in the US, regularly scheduled stress associated with that as you compete for limited resources, and then just infused chaos in there. And it's just demoralizing for people who are at the beginning of their career, people, who are participated in this study who had those resources from years ago. And so, the real danger here is that these pools of talent and sort of the inspiration that the next generation brings to the table for scientific advances starts to evaporate right before our eyes. NIH funding, so training grants, which have been under wholesale attack, especially under the guise of this, sort of knee-jerk reaction to diversity, equity, and inclusion. And so you have whole swaths of trainees who are being disinvited to the table under these sort of bogus claims about who gets what and these kind of things. And what institutes are at the table here? General medical sciences. So when we think about your point, Vincent, thinking about funding research for swordtails, you might say swordtail fish. Well, why would we think about this at a Institute of Health or something like that, including the general medical sciences? And so there are rumors circulating around now that General Medical Sciences, the institute that funds a lot of basic science, is going to sort of wash their hands of anything that involves an infectious agent, a virus, bacteria, that those will be all shunted to NIAID. And of course, NIAID, so where a lot of the core virology, other work on infectious agents should be focused on that, but so should GMS, because as we'll see today, if you're curious about the color of swordtails, you'll find yourself thinking about foamy viruses endogenized retroviruses, and all of these things are connected and intertwined. And so if we start building or putting up more barriers or building these silos, we're letting go again of the incredible bridges of bringing science together here.
I mean, there's no question that U.S. science has been a model for many countries for decades because of the diversity, the collaborations, the
Mm.
amount of money that you can do basically whatever you want as long as you can get it funded. And all that is under threat because people with some ideological views want to change it, and it's not going to work. I have a pick today that feeds into this. We'll talk about it later.
Yeah, we'll circle back to this. We're kind of putting a pin here. I'll also just highlight two other points. So, private foundations really, buoying some of this work. So here we've got the Searle Scholars Program, the Pew Biomedical Scholars Program, the Freeman Rabowski Scholars, and so this has been sort of a bright spot, I would say, in the last year or so, thinking about where science is coming from. But again, these are very limited resources, small organizations that have the courage to fund exciting and interesting work. But now we're taking sort of regularly scheduled hyper competitive or competitive U.S. science and making it hyper competitive. We can't support the scale of science, which has also been a hallmark of almost the magical contributions of science in the U.S. for decades. And so as it sort of narrows down, it starts to, people who are, invited to the table, you never know what's going to be the thing that sort of inspires the next discovery, the breakthrough that could lead to a blockbuster drug or whatever it is, just a better understanding of the world. And so we're buying fewer and fewer lottery tickets in that sense as science is being practiced in the US. And then philanthropy. So now, science is depending more and more in the US on sort of the generosity of people who have a lot of resources personally. And, that might be okay. But again, the sort of mismatch in scale here is pretty massive. And in addition, a philanthropist's interest in a topic can be kind of fleeting. So we might have, sort of venture capital billionaires who want to live until their mid 150s or something like that, but come back in a couple of years and maybe they want to develop weird drones or something. I mean, you can't, the stability here is missing. And so we've just injected across the board here different kinds levels of chaos, uncertainty, and then asked young people, incredibly skilled, talented people from all over the world to sort of trust us. Oh yeah, it'll work out somehow. But that's a pretty big ask. The farther we go here, the farther we see the damage inflicted on U.S. science. And so, some glimmers of hope here. I think some, the 2025 midterms, but this is a time to think about what's our politics as it relates to the things that we support going forward, and what do things look like in 2026 while we still have some opportunities to say, hold on, we need to stand up for some of these things that have been so fruitful and inspiring and important over the decades, all the way from understanding the world around us to coming up with medical interventions that can really aid the human condition, not just here in the US, but around the world.
I mean, this is just the key here. All this curiosity-driven research comes up with stuff that can help you. For example, all the new gene editing started with curiosity-driven research about a bacterial immune system. So you cannot have politicians just decide what work is going to be done because they have no idea. They have zero clue whatsoever.
That's right. And so on TWiEVO, I feel like, we are, that's what we're looking for, is we're sort of purveyors of curiosity as it relates to the natural world around us. And so we have a great one today. This is the topic of structural coloration, how it evolves, what's the sort of the genetic underpinnings of it, what can it teach us about some of these really interesting and surprising traits that will sort of determine whether an individual might avoid a predator or might find a mate. So what is structural coloration? So this is a lot of animals, found in a lot of animals, in the ability to produce molecules that manipulate light in order to generate the appearance of colors. And this is a little different, Vincent, than sort of just like straight on pigmentation, the way we think about, compartments and cells that get pigmented or colorful granules and things like that that can sort of influence the color of our hair, skin, various other things like this. Here, these molecules themselves aren't colorful, but they manipulate the light or reflect or refract it in ways that give the appearance of color. We've got all kinds of sparkles and shimmers that sort of underlie some of this. In a lot of animals, this happens in cells called iridophores, including fish scales, which is one of the outstanding examples. So if you're taking a break here in the U.S. from the holiday turkey tomorrow, go to Google and type in zebrafish scale image or electron micrograph, and you'll be blown away. Like we look at the size, or we look at, with our eyes at these scales, you can kind of see these shimmery patterns as you zoom in. It is just, this is nature's artwork here, seeing how iridophores can play with the light in ways that sort of create these effects that could be adaptive in some cases. And so we'll kind of go all the way from sort of the process here, the genetics, and then into some thinking about, well, what might be going on in terms of the adaptation.
So this is structural coloration is different from pigment-driven coloration, right?
Exactly. So the pigments themselves, right, have that are various colors. Here, the iridophores, and actually in the case of, we'll get into the weeds a little bit more here, but in these cases, the iridophores, so these cells have stacked platelets, sort of modified platelets within them that are playing around with the light. And so they themselves don't have a specific color, but that's what gives it in that sort of shimmery appearance, depending on how the light is playing with the iridophore. It might be sort of purplish or silvery, or various colors across the visible spectrum that at least our eyes can detect. In other animals that have that wider range of probably picking up even more of the manipulation of the light. Okay, so that's kind of the topic here, is structural coloration. How might it evolve in different ways in different locations in different animals? And the protagonist here is the swordtail, or our protagonists are both the swordtail fish that have this trait in some cases. Swordtails, I think most of us are probably familiar with. If you go to your local pet store, you'll see some of these finny critters swimming around. So and if you look at many of them, males in particular, females in some cases, you have these really extended, my fish, we'll see if my fish morphology is good here. The caudal fin, or the tail fin, I should say, sorry. The rear fin, tail fin, extended, which gives the swordtail sort of its appearance in its name. And so
these are fish that you would have in your aquarium, right, Nels?
Absolutely. These are also fish that you find in Mexico, the Veracruz region, southern Mexico. And so the Schumer Lab and others, to sort of kick off this collaboration, we're doing some field work in collecting swordtails from the wild, and noticed in a population that hasn't been studied, some that have this quality of shimmering sort of across the side of the fish near the lateral line. Yeah, we got to show some pictures of these fish. Yeah, absolutely. So if we look here, yeah, perfect. So we've got a phylogenetic tree of some of these species. You can see at the top there, one of those swordtails in the tail fin. And then, down the fourth fish down of these close relatives of swordtails, we have the sort of non-sparkle variety that was pretty familiar. And then this new sighting in the wild of the sparkle variety. And so if you look sort of from the dorsal fin back towards the tail, there's this extra area that has this sort of silvery shimmer. And that you can, as you zoom in, that traces to the scales, and the iridophores, the cells that are associated with the scales in that region of the fish. So they call that trait sparkle. And that's in contrast. So if we go down to the sort of third fish that zoomed in on, to this subdermal irradiance. And so that's happening, I believe, below the scales. It has been observed before, but this sparkle phenotype here out at the surface in the backside of the fish, that's a new observation for this family of swordtail fish. And so that's sort of the, that's a peek under the hood here at sort of the cool observation from the field. And then the second protagonist that's going to arrive, there's going to be an endogenized retrovirus um, that's
Mm.
activity, just its selfish replication, turns out to be responsible for that difference between non-sparkle and sparkle. And so, Vincent, we've talked about the impact of transposable elements and related sort of endogenized retroviruses on TWiEVO, on TWiV for many years. And you can kind of think about these viruses as mutagens. So as they're jumping around in genomes, selfishly replicating, sort of in the framework of Richard Dawkins, you have this insertional mutagenesis where a new virus sequence or transposable element sequence will land in a different part of the genome. And that can have some impacts on the in the host genome, on gene regulation, and other biology. So we kind of got this on the record early in TWiEVO's history. TWiEVO number 10, spicing
Hmm.
up peppered moths with a selfish gene. So this is heading over to England. Some of our viewers there, Barb Mack, others, the peppered moths. So this interesting sort of collision between air pollution and the ability of predators to hunt for moths. The peppered moth arriving as an insertional mutagenesis event that changed the pigment of those moths. So this isn't sort of totally new science. There's some echoes even in some of the TWiEVO episodes before. Vincent, TWiEVO 36, all's not quiet on the telomeric front. We talked to one of my good science buddies, Mia Levine, who runs her lab at Penn. She's talking about the telomeres of Drosophila, which are not just these short 7 nucleotide repetitive regions, but are actually dictated by the activity of transposable elements that hop and are retained out at the ends of chromosomes, the telomeres, the telomeric front, to maintain chromosomes so they don't erode, so that chromosomes have this level of stability. Totally depends on the activity of these transposable elements. We had a couple of crossovers over the years. TWiV 382, one that's near and dear to my heart. You talked to a former postdoc and me about everyone's a little bit viral. So this was Ed Chung. He was when he was here at Utah co-advised by me and Cedric Fischott, and is now, I'm happy to report, running a massively successful lab at University of Colorado Boulder. Ed's still on the case here. So in this case, he found that ERVs, these endogenous retroviruses that will be the topic of the swordtails, in our genomes, in their propagation, have a role of impacting the regulation of our immune genes, our so-called interferon stimulated genes, or ISGs. And this is wild, man. So it's like a endogenized retrovirus, something that's over-replicating or selfishly replicating as a virus, is actually contributing to our own immune regulation today. And so these sort of back and forth, almost arms race scenarios. So Ed
Makes
is,
sense because in the retrovirus you have preformed gene expression cassettes, right? The LTRs. And so rather
indeed.
than the cell making its own, which could take a while, one pops in and you just use it, right?
Absolutely right. And we'll be back to those LTRs in a little bit as we think about the foamy virus of swordtail fish. But I can't resist to do one more, sort of trolling through the archives of TWiEVO and TWiV. This is a more recent one, TWiV 935, mobile DNA with Cedric Fischott. So this was Ed's second mentor when Cedric was here. Again, so listen to this podcast. I mean, Cedric is just a champion of mobile elements and how they've sculpted evolutions of hosts, since time immemorial. I'm holding up Cedric now as, an example of major league science in the U.S. So Cedric opened his lab. He's from France, originally grew up in France, opened up his lab at UT Arlington in Texas, moved here to Utah when we had our labs just across the hallway. Some incredibly fun years of working together, collaborating with great and talented postdocs like Ed Chung. And then Cedric then moved on and is now running his lab at Cornell University. And so again, this just sort of fluidity of American science where people from all over the world come, train in places like Texas, Utah, New York, and then just champion sort of this understanding of the biology around us as it ranges from our genomes and the genetic elements, the transposable elements, out to the retroviruses, and all of that impact on the biology that we see, including with our own eyes.
That was
Okay,
one
sorry.
where I went to Cornell and interviewed Cedric, right?
Yeah,
Yeah.
yeah.
I
Isn't
always
that
wanted to
fun?
do
Just
that.
hanging out with him.
Well,
Yeah,
he
he's
can
just
talk about anything. It's great. And when you ask him a question, he can just go on and explain it so well. And then you come up with 20 more questions. It's just so much fun to talk to somebody like that, right?
couldn't agree more. And just that sort of motion of the that
Kind of
he
like you, Nels.
breathes into the science. Well, I'm trying to, to do what I can.
No, but Nels,
Um,
if I
yeah,
ask you a question, you
yeah.
answer it, and it's interesting, and it makes for more questions. This is not everybody does that, right? But when you do, it makes it for a pleasurable learning experience. That's the point.
Oh,
And
absolutely. Fair enough.
I'm not
And
just saying that because you're on Microbe TV podcast, but because you have the talent, dude.
well, well, thank you. But Vincent, I have to on the sort of holding up the mirror to you as well. Your ability to draw this information out of scientists who, honestly, many of us would rather be sort of hiding under a rock, you know,
Right.
hanging out in the lab, just thinking about our things. Your ability to draw people out and to share the science that they do and why it's important, why it's interesting, why it's fun. That's also, I would say, a very rare trait, which is why it's so fun to get together once a month and do what we do on
Well,
TWiEVO and to, yeah,
the key now is to be curious, right?
yeah, it helps. And then to to get your courage up, gain some experience to just talk about it.
Well,
And
I
yeah,
think Microbe TV is a good platform for that because you come on, we are non-aggressive questioners, right?
yeah,
We just start by saying, so Nels, where'd you come from? Where were you born? And you're like, I can answer that question, and then we
yes,
slowly ease you in, and
yeah,
it's not a problem, right? We never ask you questions that are aggressive or confrontational. We just ask you about the science, right? And
yeah.
that's why we can bring it out in people, I think.
I agree. Baby steps, and then and trying to build on that. Not only to share the science, but to also try to defend or to stand up for it, as we've already been spending a lot of time discussing. Okay, so that kind of sets the table. So we're going to pick up this story with with an endogenized retrovirus, and I guess sort of getting to the punchline. Um, this retrovirus is sort of has the features of both a transposable element. So it's endogenized in the sense that retroviruses, we should say, in their replication cycle, integrate by reverse transcription from RNA to DNA. They insert themselves into the genome of the host. And then for exogenous retroviruses, they get back out. They encode an envelope gene so that as they replicate, transcribe from that DNA copy back to an RNA copy, they get packaged in an envelope and then can engage some of the cellular machinery to get out of the cell to go on to infect other cells, or then pass to other hosts to transmit. A lot of endogenized retroviruses, we catch them kind of in snapshots, evolutionary snapshots at different sort of moments in their history. And so a lot of times these exogenous ones that might be passed horizontally from host to host lose some of the machinery or the genetic codes for the envelope or other components of it, and in a sense get marooned in the genome of the host, but can still over replicate. They can still be active. And then as they're just like a bull in the china shop running around inserting in different parts of the genome, that can have an impact. And one of those impacts, Vincent, you were already hinting at. What are these long terminal repeats or LTRs?
These LTRs are the sequences at the either end of the provirus, the integrated viral DNA, which contain
Mm-hmm.
promoters, enhancers, transcription factor binding elements. So they drive expression of the provirus, and then the the right hand element stops it. But of course, it also has the same promoter, so it could turn on downstream genes as well. And that's the basis of retrovirus mediated oncogenesis in some cases, right?
Yeah, exactly. So oncogenesis in some cases, or anything that as you're messing with transcription of genes nearby, where that sort of insult or that insertional mutagenesis has happened, and that could be if it's something that upregulates an oncogene or downregulates a tumor suppressor gene, upregulates a developmental pathway, as we'll see today in the swordtail fish. This could have a massive impact that then natural selection might act on, depending on what the outcome or the phenotype is of this genetic variation.
So
Okay,
one of my
so
favorite examples is
mm,
blue chicken eggs, right? The blue
Yeah.
in a chicken egg is highly prized, so farmers breed their chickens so that they make blue eggs because people like to eat blue eggs. And the blue is made by a pigment. And it turns out that what turns on the pigment gene is a retrovirus inserting upstream of it. And so
Yeah.
that's what the farmers are breeding for, that endogenous retrovirus element there. But I love it. I think
Exactly.
it's a harmless effect, right?
Hmm.
It's very pretty, and it just is a great example of how moving genes can turn on or turn off other genes.
Couldn't agree more. And in that case, a case of conscious selection, right? The farmers or
Right.
the hobbyists that said, "Oh, those blue eggs are interesting. Let's keep them." And then that's the selective pressure. And then, as geneticists, we might then say, "Okay, well, what was it in those chickens that made those eggs blue?" And then you find yourself with a virus on your hands, right? And
Yep.
so it's right. It's those sort of hidden connections, and then asking how often is it a virus or a transposable element? And the answer is quite a bit, right? So those peppered moths, there's sort of a laundry growing list. And when you sit down with someone like Cedric Fachat, who can just tell these stories one after another, you start to get a sense that, hold on, who's in the sort of biological driver's seat? Is it us, or is it the viruses and these elements that are sort of chiseling away on genomes and then when you add selection, whether it's conscious or unconscious, maybe it's a predator or a mate or gaining one, etc., that can influence this, or to come.
Now,
Yeah, yeah,
now something I've always thought about.
yeah. Mm.
Does the moving around of an endogenous retroviral element provide any benefit to the retrovirus, or is it just a random outcome of the way the DNA is constructed? So is it good to be able to move around the genome?
I think the answer is yes, but it's always some of both, right? So if we think about this as a mutagen,
Mm-hmm.
as mutagenesis, there can be ones that are negative, both for the
Sure.
host or for the virus. Ones that are neutral. So it's just like, sort of genetic noise in a sense. These things are over replicating or jumping around, but not having a
Mm.
fitness impact or adaptive. And so, this gets at sort of echoes on this notion of exogenous versus endogenous. So let's
Yeah.
say it's a transposable element now that has no possibility of getting out of the host genome on its own, right? It can
Mm-hmm.
it can go through the germline, and so through vertical transmission from parent to offspring, that can be passed, and if those parents or if those offsprings, right, or offsprings, if those individuals are adapt, like if there's a change caused by a transposable element, that blue egg, for example, and then you
Mm.
breed tons of chickens, now that hitchhiking along, that transposable element will sort of sweep through the population if we select only on blue egg layers. So that's one way that overreplication can propagate. But then there's another great one, and this is sort of a topic that we think about in my lab, which is what if these elements team up? So if a transposable element is replicating, has this sort of genetic energy to it, but now an exogenous virus, my favorites are the things like pox viruses, herpes viruses that encode massive DNA genomes relative to some of the more streamlined retroviruses like the one we'll be talking about today, a foamy virus. As they're replicating, if there's TEs hopping around, they might not only sort of hop into the host genome to get more copies, but hop into that
Mm.
virus genome. And now, as that virus completes its replication cycle, goes on to infect other hosts, now that TE got a hitchhike to free ride to a whole new, to be horizontally transferred to other individuals, even other species. And so, again, this is sort of these conduits of genetic communication, the highways of genetic exchange of
Mm.
information that I think underlies the diversity of life around us. And so I think we're just glimpsing at sort of, these kind of events and the massive impact that viruses have on moving genetic information around so that can be kind of road tested by trial and error to see what works. And then that's sort of the fuel or the engine of the diversity that we see. So that's, kind of a hypothesis. How much of the tip of the iceberg are we seeing? Are these special cases, or is this sort of the language of biodiversity? I think that's an open question for the field. That's one that keeps me up at night or gets me back
Mm.
into the office in the morning is really thinking about stuff like that.
So Nels, You work on zebrafish. You work on a little fish yourself, right? Do they have sparkles?
Guilty as charged. They do, and actually, I think we'll get back to that in a moment. But
All right.
Vincent, let's go down to Veracruz, Mexico, and a field trip with the authors of this work near Juntas Chicas in Veracruz. So they were out collecting swordtails. Molly Schumer's lab, is becoming increasingly famous for finding these really cool, color morphs or,
Mm.
some of that diversity that you showed in that slide from Figure 1, and then building around, what does this mean for mating choice and things like that, or avoiding predators, et cetera, and drilling all the way down to a genetic understanding. So anyway, they're out doing a collecting trip and find this unstudied population of swordtails, and notice about a quarter of them have that as reflective scales, the trait that they call sparkle. And so if you look under the EM, they have these stacked reflective platelets associated with the iridophores, and the ones that have that sparkle trait have a different shape than the ones that don't. And so here's immediately where your question about zebrafish arrives. So zebrafish also have iridophores in their scales, and because developmental biologists for 50 years have spent a lot of resources to understand how these iridophores work, et cetera, there's this base knowledge that the swordtail community can draw on to immediately sort of, add rocket fuel to advancing the work in these wild species where you don't have all these genetic tools. And so, to pause here, I think there's just this great bridge forming between kind of the massive revolution of molecular biology in the last 50 years, some of the tools like CRISPR that you were mentioning, et cetera, and all of these accessible systems, and bringing that now to the rivers and tributaries of Veracruz, Mexico, right, to the natural biology and the complexity that we see all around us, and to try to use those model genetic systems, all those tools, to sort of, advance or build off all of that knowledge. And it really can accelerate our understanding. In a lot of cases, these ecologically threatened habitats, to try to sort of a race against time to understand or reveal some of the exciting biology around us that might otherwise be lost. Okay, so so they immediately could form some, I think, useful hypotheses about what might be going on here with their sparkle trait as it related to what's already known about the genes and developmental pathways of iridophores that have been studied for decades in zebrafish.
One thing that's worth pointing out is that this sparkle trait appears before pigment-based traits would appear in the fish, right?
Very important. Yeah, sort of putting together the developmental pathway. And so looking at or hatching swordtails, bringing them from the field into the lab, hatching them and watching them develop in exactly your point, seeing that, okay, wait a minute, this probably isn't going to be a pigment story. And instead it draws on the iridophores. And the EMs that they did with some of their German collaborators supported that direction as well. Okay, so then as a geneticist, the next, I think, sort of burning question is, okay, well, what's the genetics underneath this? How do these things sparkle, especially when you see a quarter, like immediately your sort of Gregor Mendel radar should peak what's like a recessive trait, right, or a dominant trait, and thinking about that. So they wanted to find the genetic basis of why are a quarter of these guys sparkling. And so they sequenced the genomes of 260 individuals of the population and did genome-wide association studies, or GWAS. So they took 66 sparkler individuals, their genome information, 194 non-sparklers as controls, and then they ask, what are the differences in that comparison that associate with that trait? Is there something about the sparklers' genomes or a region of the genomes that is different from consistently versus the non-sparklers? And that's a GWAS. And you use statistics to try to really pinpoint the regions that might be at play here. And there's this great figure in a lot of GWAS, especially this one was a really clear one, a Manhattan plot. So for each of the chromosomes, you go along, and it sort of looks like skyscrapers of peaks where there might be an association. The higher the building in the Manhattan plot, the more likely you have a true association based on statistical testing. and they had one skyscraper there. I think it was on chromosome 15.
Yeah, we'll show that here. It's
Yeah.
beautiful right here.
There you go. Yeah. There it is. Yeah. Panel A at the top left there. And so each of those dots is a genomic region. And on the y axis, going from bottom to top, is a statistical score. And as you can see on chromosome 15, there's the Empire State Building in Manhattan looming over the rest of the lower ones. It's not always that clean. If there's multiple traits at play, you could have all kind, like sort of decoding the sort of complexity of the New York skyline relative here to. I guess this is more like a Manhattan, Kansas plot where there's probably one tall building. it's pretty clear the other Manhattan in the Midwest. Okay, so that meant with that information, basically they could narrow their candidate genetic region, explaining the sparkle trait to 400 kilobases. That's 400,000 ATs, Cs, and Gs, which encoded for 16 genes. So this is really common for GWAS. You sort of narrow the playing field, but you still have like a lot of kind of competitors, so to speak, in the race of what are the genetic variation that underlie a trait. So what do they do next? They used more sequencing. So this is, again, in the post-genomic era, the ability to just like sequence a ton of stuff into the hundreds at the genome level, whether it's DNA or RNA sequences. What are the differences in genes that are turned on or off, upregulated or downregulated? And so they did bulk RNA sequencing from scales, the iridophore cells associated with the scales, from sparklers and non-sparklers, and they found 91 differentially expressed genes. So in those sparklers, there were 91 genes that were either turned up or turned down relative to the non-sparklers. 2 of those 91 genes were in that 400 kilobase region. So they're kind of, narrowing down using different lines of evidence to propose what is the genetic change or difference between the sparklers or non-sparklers. Those 2 genes, Vincent, one is called SMYD2, SMYD2. The other is called ELCL2A. We're in alphabet soup territory as usual as we move from sequenced gene names. And ELCL2 is a known pigmentation gene that was closest to that GWAS peak, the sort of top of the building in that Manhattan plot that we looked at. And so again, this is how, without all of that work by developmental biologists, all of the work in the field on pigmentation, like it just is a shortcut to getting to like an understanding of something. And so, again, the value of basic science over the decades just echoes on and becomes even more powerful the more we have these foundations, or we can move really quickly to learn new things about the world around us. Okay, so what is this ELCL2A gene that sort of is singing out as something that's connected to the phenotype? So that gene encodes a protein, and it activates tyrosine, receptor tyrosine kinases in the so-called ELK family. In zebrafish, that contributes to the development of iridophores. So again, just what I'm saying, right? All of that investment in zebrafish as a monogenetic system of developmental biology pays off for the swordtail biologists here, who are the evolutionists who are really curious about this trait. So they were definitely pointing in the right direction. They weren't totally convinced. As good scientists, you want to get multiple sort of different lines of evidence. They started doing quantitative PCR, so to amplify this gene, specifically in the sparklers, and compare that, to at the right sort of developmental time point that they observed, and they see that gene is overexpressed using this quantitative PCR. They see more copies of it in the sparklers at that time point relative to the non-sparklers. And among those 90 genes are other genes that are involved in iridophore biology. So it's all this kind of circumstantial evidence that sort of builds. Okay, so that kind of got them to like pinpointing in the genome on chromosome 15, sort of a difference, genetic difference that probably underlies this, and it's pretty strong evidence. So then they wanted to know, okay, well, what are those differences? Like if we kind of fine map or really sequence over this. So they do sequencing on chromosome 15 and assembled 11 different haplotypes. So these are sort of collections of genetic variation from one individual, kind of in this case across chromosome 15. And as they compare these distinct haplotypes, there's one thing in common found in sparklers that's missing and non-sparklers. And that's a 17 kilobase insertion downstream of that ELK2A gene. So it's actually, turns out it's in the intron of a neighboring gene. This is sort of an interesting sort of going down the rabbit hole of GWAS complexity. The gene that might be getting effective isn't necessarily the one that has the genetic difference, because you have, as you're hinting at, these retroviruses or transposable elements can be enhancers. They can affect or the transcription of neighboring genes nearby. And so that's actually, this other gene that was next door, they tested that and saw that the insertion here didn't have an effect, but that would have been sort of another candidate that they sort of crossed off the list here. And the sparklers, turn out, all have at least one copy of this insertion, while the non-sparkling swordtails don't. The gene itself, the alkyl 2A gene, is identical. This sort of hints at like an ongoing question in the field of evolutionary biology, which is, do changes in gene regulation dictate sort of adaptive advances or leaps, or is it changes in the coding region, the gene itself, that is adaptive? And the answer is for some of both, with definite contributions to things like regulation, which is where this story sort of was pointing. So what is this insertion? And here we're find ourselves in a TWiEVO-TWiV crossover. This is an endogenous foamy-virus, a retrovirus. Vincent, remind us or tell us about what these foamy viruses are.
So foamy viruses are so named because when you infect cells in culture with them, they give the cells a foamy appearance, right? It's the kind of cytopathic effect. It's a typical retrovirus with an RNA genome, but although a certain fraction of the time the genome is DNA. It's kind of like hepatitis B virus, where the genome is DNA. Because what happens is these retroviruses are assembled in cells, and then your usual retroviruses are released as RNA-containing particles. But for hepatitis B virus and a certain fraction of foamy viruses, reverse transcription commences in the particle before it's released from the cell. So you can have different amounts of DNA in the particles. But they're bona fide retroviruses, just a different class compared to the ones we usually talk about.
Yeah, exactly. And remind me, do foamy viruses infect humans?
I don't think they do. That's a good question. Let's look it up.
Let's look it up. I think it's pretty rare. So, our closest relative, there's a simian foamy virus that's known to infect primates. That's probably the closest one currently that might impinge.
So we can get them from non-human primates if they bite us, right? Yeah.
Yeah, and so they're right. So those rare crossover spillovers, right?
They don't seem to cause disease, but they stay with you because they integrate, right? So you shouldn't get bit by a non-human primate, Nels.
I think that's always practical advice. Whether there's a foamy virus there,
zebrafish.
Guilty as charged. That's right. Although, so here what we're learning for the first time is the impact of foamy viruses in fish. So I shouldn't say for the first time. So in fish in general, but for this specific family, for swordtails, and then the surprising link to this sparkle pattern on the surface of the fish. So, as evolutionists, they do a good job of starting to try to catalog some of that foamy virus diversity here, and to start to ask. So, this one, and we'll kind of go into the weeds here a little bit. That's that topic we've discussed: the difference between an exogenous retrovirus, one that can replicate, get into a cell, replicate, get out of a cell, and even get to another host, versus an endogenous one that has lost the ability through mutation, sort of erosion of the genes that encode the envelope or other genes that are needed for it to get back out of the cell. So they propose that this one is an endogenous one, but it's endogenized, but that's very recent. And in fact, they can see an intact, the LTRs that we'll talk about in terms of the impact on gene regulation, GAG, POL. So these are sort of the key genes that encode the proteins for the reverse transcription, the polymerase, the GAG, for the sort of the business end of the virus, and then the envelope gene, which coats the RNA to move forward. They call this ERV-F.1-XEVE-chromosome-15. That's the current name of this virus. I have to say, Vincent, this is a little bit alphabet soup, not super inspiring. I would have probably named it Sparkle Pony.
Yeah,
as
that
a
would be great. I
I
love it.
mean, yeah. So, that's one of those fun things with discovery science, right? Where if you discover something, you get to name it. Um, they're doing, a careful job, I would say, of a useful title as it relates to its relationship to its other family members. But just like they named that sparkle trait in the swordtails, I couldn't resist calling this virus Sparkle Pony. Okay. Um,
Because, as an example, I mean, this kind of quirky naming is very
yeah.
common in fruit flies, sometimes in
Yeah.
yeast. And one of the transposons, one of the DNA transposons, is called Sleeping Beauty. It's
There you
great because
go.
it was not moving around I think it was a fish genome,
That's a
and then
zebrafish.
zebrafish, and they fixed it, and it woke it up. Sleeping Beauty.
There you go, Perry Hackett. So there is, yeah, branding matters in science, right? To have that sort of memorable connection, and in the beauty is you can name them both, right? It can have the sort of formal name as it relates to its other relatives, but you can also be a little fanciful there. So
Okay, Tom says his cat is named Sparkle.
oh wow, here we
Granddaughter
go.
chose the name. So
There you
somebody
go.
needs to call their animal Sparkle Pony, and then you will have validated Nels.
I know. So Mike, I should confess here, I'm the father of a kindergartner who's super into unicorns and, sparkling horses and stuff like that. So it's sort of at top of the mind for me in this case. Yeah, that's great, Tom. I love it. Sparkle the cat. Okay, so this is a foamy virus. So a family that's common in swordtails, other fish. And immediately, one of these examples of the gifts of infection, right? We've got an entire sort of narrative out there that viruses are negative, they make you sick. Entire podcast devoted to viruses, to the kind that make you sick. But as we continue to explore and study, we can also see that these viruses can also have impacts that are actually really gain of function impacts, so to speak, if I can reclaim that title of bringing traits to the host. And here it's looking pretty convincing that sparkle phenotype or that trait in swordtails is the result of the action of a virus. Okay, so they end up doing, I think a good job of starting to to understand where this virus came from and what it looks like. So they did sequencing on, I think, 10 additional foamy viruses that they found actually at 18 different genomic locations. So any self-respecting endogenous retrovirus doesn't just land once and get out of the game. They can find it distributed. Depending on the individual that they look at, they can find 4 to 11 additional foamy virus genomes or parts of them. A lot of polymorphisms here. And so this gets at the idea, that this insertion was a relatively recent event, and that it's possible that these foamy viruses are still on the prowl, that they're still moving around the genomes and maybe they'll impact another trait that natural selection might act on. Consistent with that is they get RNA of foamy virus RNA from their bulk RNA sampling, and they see that in some of the tissues, including the male germline. And so that also gets at sort of the vertical inheritance, or how these things, even though they're endogenized, might move. So they do some phylogenetics, as a self-respecting evolutionary lab would do. And the foamy viruses of swordtails, it's messy. A lot of signs of host switching. And this is kind of gets at what we were talking about, Vincent. These, the notion that these elements, retroviruses, are being swapped around like virus trading cards between hosts, and that this is sort of part of the underlying action of evolution here, as selection might act on it.
Trading cards. I love it. It's great.
Yeah, right. I mean, this is like, and
Yeah.
if you're trading cards, you want like a baseball card, right? You might have the nickname of the baseball player or the stats on the back or whatever. Here, I think Sparkle Pony. I'm lobbying for Sparkle Pony. It's still a preprint, so maybe in peer review, who knows what could happen.
They're going to hear this podcast and go, "That's a great idea." And they'll thank you in the acknowledgments.
Let's not hold our breath on that one, but but we'll see what happens. Keep following this story. Keep following Molly's lab.
These are endogenous retroviruses, so they're passed on to offspring through the germline.
Right.
But the original infection, how did that happen? Now, it's just virus floating around in the water, you think?
Well, yeah, possibly. And then it comes online, and that's a pretty interesting question too, that population genetics can say something about, So they went back to the virus. They do a really cool job sort of going between host and virus as the manuscript continues, as the preprint continues, and are curious whether natural selection is acting on that region. Is this an adaptive trait? So they do two kinds of tests, population genetic tests. The first is to ask, okay, are we just looking at, is this really trading, exchanging trading cards, or is this just noise, right? And so they end up by sequencing the virus region from a number of individuals, they can look for these signals, do these genetic tests to look for signals of selection. And in fact, they have, it's not definitive, but they have at least some weak signals of balancing selection. It looks like once this thing arrived, that's being maintained. Balancing selection is sort of a case where the most famous case is probably like sickle cell anemia, right? Where you have, if you're a carrier of the trait, you enjoy resistance, in this case, to malaria. But if you have both copies, there's a trade-off or detriment, which is you have sickle cells, and you have you're still resistant to malaria, but now your blood cell biology is working against you. So they see maybe a glimmer of that for this, and what that means, I think, is work ahead. But then getting closer to your question, they're able to try to date when this thing arrived in the genome. Based on the population genetics, the variation that they see in the sort of the patterns of the genetic changes. And so they propose that about 16 to 20,000 generations ago, and I don't at my fingertips know how long a swordtail generation is, maybe 3 months, I'm guessing, but so multiply that by 20,000, and that might have been when an exogenous foamy virus, the sort of ancestral virus, arrived, inserted into the genome at that location. And then there could have been all kinds of fates after that, right? It could have been a rare event where any individual that had that sort of insertion that caused the sparkle phenotype, maybe maybe actually they were more apparent to predators, all got picked off, and then that would have been lost. Or it was maintained for some reason. There was some adaptive potential. And so, I think this is a great moment maybe to sort of pause and just like look out across the horizon and remember that the amount of sort of trading card attempts here, but with viruses, is probably off the charts. The ones that stick around for 20,000 generations are quite rare. And so in that time, there are probably countless foamy virus insertions in various populations. And the ones that are still here today are these sort of rare ones like Sparkle Pony. The virus that they see here. Sorry, I've
So
no
you
really
think
told you.
there's some selection in this case? For here, we're going to talk about this later. That this
Yeah,
Sparkle
yeah,
may
yeah.
have a selection, but
Right.
there's certainly many endogenous retroviruses that just sit there because they're no harm, right?
Correct. And so you might find them in various stages of disrepair.
Yeah.
However, they might persist that way. Or it might not be something about hitchhiking with the host. So this one, where sort of the authors are proposing it's sort of a gift from the virus that turns on
Sure.
the ELK gene that sort of then promotes the shape of the platelets in the iridophores that then have the iridescence. But there are other cases where it might just be an advantage for the virus, right? It's over replicating. If that means there's other sort of accomplice viruses, the pox viruses, giant viruses that you see in aquatic environments that are sort of gene thieves that are hunting, like that's another, just persisting then might mean being passed regardless of any sort of gift that it gives to the host, or even sort of adaptive potential itself. And so, yeah, there's all kinds of possibilities here, which I think, again, points to the work ahead, how little we know, but how much is possible in terms of sort of the genetic underpinnings of how viruses replicating or over replicating can really have all of these different outcomes.
So here's a tough one. So
Yeah,
the driving selection on viruses is to transmit to a new host, right?
agree.
That's really in the end. So if you infect a host and endogenize, are you successful?
Well, possibly. So if you have that sort of vertical inheritance, can be super successful, right? And honestly, the viruses don't care, right? So this is just what happens. And then whether it persists is sort of out of the hands of the virus or any genetic element. That's evolution playing out and sort of the seesawing of forces. And it could be, in the history of sparkle are other things that didn't make it, that we can't see because we either we haven't um, looked at enough biodiversity, or it was there but is missing. It could have been super adaptive in a few individuals, but then like a smallmouth bass came over or something and just ate them all, and so it like had that potential, but there was just this sort of contingency, an event sort of that doesn't necessarily relate directly to this. Could have been an extinction event, or let's say another virus actually. So the endogenized virus might provide this really cool fitness advantage for avoiding predators, and we'll sort of look at what they have to say about that in a moment. But if that ability to avoid a predator is sort of overwritten by even like another virus that might have like had fitness, like as it was replicating, caused the extinction of that population, then it's sort of like that virus won out over the other one. And so, yeah, there's all kinds of population level dynamics that might be at play in
So
these cases.
the thing is that retroviral integration is an obligate part of the virus reproduction cycle. So in other words, in order to make new particles to spread to another host, it has to integrate. And so for a period of time after integration, these integrated copies are making viruses. And as you said, they could spread to other fish, presumably. They could be excreted and spread to other fish. When they undergo mutations as part of replicating in the genome, which silences them, then it's a failed transmission because it no longer makes particles. And then it's no longer part of the retroviral reproduction cycle. It's part of the host at that point. That's how I would view it.
It switched teams in a sense, or
Yes,
it's become
exactly.
become dependent on that team. And by the way, there's all kinds of incredibly cool stories in other populations, not just swordtails. So there's active ERVs and koalas. I think there's some active ERVs in deer populations, like in Montana, in the Mountain West,
and rodents too. Yeah,
rodents as well. And so, by having these sort of different points on the map and different hosts and different species, again, the value, that sort of curiosity driven, what's going on with this one? And then you can start to compare them to each other and start asking, well, are there sort of shared principles of what works or what doesn't work to get at the rule book of these things? And so it's not just the swordtails in Veracruz, Mexico that matter. It's the koalas in Australia, and it's the deer and rodents in the Mountain West. And so that's where these sort of interdisciplinary, this work from all of these fields, genetics, evolution, field ecology, et cetera, is really, I mean, that's to me kind of underlines the value of this. Okay, but let's get back to the field, Vincent. So what is this sparkle trait good for, if anything? And so it's certainly caught the attention of these scientists, of these evolutionary biologists, evolutionary ecologists, but is there something about sparkle that might, so if we come back in 100,000 years, will every one of these fish in that population be sparkling? And so they think about the two kind of obvious fitness outcomes or influencers here, and it's between mating preferences. So if you're a female or a male that sparkles, does that mean your mate might be like, hey, that's someone who I'd like to have kids with? Or does this impact predatory responses? And you could imagine that going in either direction, right? So if you're sort of conspicuous, a predator might say, hey, there's a meal. Or there's these interesting ideas that, if you sparkle, you might startle or confuse a predator. And so that's what they're going to end up pointing towards here. So the predators that matter for this population of swordtails actually aren't bigger fish. It's birds that are flying around and scooping them up or eating them. But they've actually, I should say, before we get to that, they ruled out some of the obvious mating preference stuff. So they did some tank trials to test whether there are differences in boldness or mating attractiveness. So that between tanks of all sparklers or non-sparklers, or mixing them together, and they don't see any differences that they can link to or correlate to having that sparkle trait. And so then they moved to predator evasion, and they point out that in videos of these sparkle fish swimming, there's a flash as they change directions. So as the fish is, making a sharp turn, that's when the light hits the iridophores with the aid of that foamy-virus, endogenized foamy-virus, or potentially active virus, to give a flash from above, right? So the light comes down, reflects back up as you look at this video footage. And so that kind of got them onto the idea of maybe that could confuse, the birds that are preying on the fish. And so to kind of start to test this, I would say they haven't like put a nail in the coffin of this sort of fitness outcome, but they built some model birds and swooped them across the tank, and then asked what the fish would do. And so there's a startle response, right? So they change directions, and and both fish do that, so you don't have to sparkle to try to evade the predator. But in about a quarter of those sparkle fish, they see a flash, and so that's sort of the next question, I think, where the paper wraps up. In this case, is to say, is that flash something that then means that you're less likely to be a meal for a bird? And there's some obvious experiments. I don't think, some high degree of difficulty, but obvious experiments that you might do to try to see if there's evidence for that hypothesis.
Yeah, you could knock out the gene and put the fish in a stream and see what happens, right?
Indeed. And so easier said than done. So this is an unstudied population of swordtails. So the ability to delete that gene, I think they mentioned that it's not yet within grasp, but that's on their menu. And they're, I had a really fun lunch with Molly Schumer a few weeks ago, which is why this got on my this preprint got on my TWiEVO radar. And we were sort of daydreaming or, brainstorming about advancing these kind of things. Should it be in a stream, or can we recreate some of these? They did a model bird, but how sort of extravagant can you become in sort of creating this in controlled settings where you can control variables? I think there's some really fun opportunities to think about recreating, rivers or riverines, tributary kind of at science museums or science buildings, and then bringing some of this complexity into sort of, into our own sort of scientific frameworks. That could be another really cool.
The reason I mentioned a stream is because some of the earliest experiments to understand why sex arose were done in organisms put in a stream, and they fence it off so they stay in one place, right? So it's a natural environment. And they saw that when the organisms had sex, they were more resistant to parasites.
There you go.
And I think there was an organism that could do both, and so you could really get good confidence in the data. I forgot what organism it was.
Yeah, me too. But you're absolutely right. There's also, experimental evolution. So not swordtails, but field ecologists have taken guppies, some that are highly colored, some that are less colored, mixed them all together, put them in ponds. I think this is in Central America. Right. And then either in the variable they're controlling here out in the field is either having predators or no predators, let the populations go for months or years. And then if you come back, there's some of the patterns that emerge are that the ones without predators become more showy and colorful, whereas the other, this is pigment-based morphology, not iridophores, iridescence, but and the ones that have predators become more drab. And so there's that sort of, you're litigating sort of the mate attractiveness versus the predators.
So if you're drab, you're not noticed as much, right?
That's the idea. Yeah. It gets more complicated, and there's sort of twists and turns. And then, like you're pointing out, Vincent, like the role of mating itself for avoiding predators, for avoiding parasites or viruses is also, it's all kind of wound together and intertwined.
You can easily get fooled, right? You got to be careful.
Yeah. Oh yeah. And then getting fooled, mimicry, right? That sort of moves in as well. Another sort of classic tie to the field. So the early naturalists from England that came over and observed butterflies that looked like each other, one's poisonous, one's edible, Batesian mimicry. Yep. Identified or inspired through work in the Brazilian rainforest hundreds of years ago. And so, what signal is true? What's misinformation? Biological misinformation or disinformation? All of these things percolating right under our noses.
Very cool.
Agree. So fun. I can't wait to see where the work goes. moving forward. And if you're looking for a lab to watch, Molly Schumer's is one to keep your eye on. They're doing exciting things, bringing together all of these fields, people from around the world to do really exciting evolutionary biology work moving forward. I guess do we have some questions? I've still
Yeah,
got
let
a
me
couple of
pull up.
axes to grind, but yeah,
Let,
yeah, yeah.
let me pull up. So Tom writes,
Hmm.
domestication of a retrovirus to a useful rather than a damaging event, which can also happen. So that's the whole
Yeah,
theme of this paper for sure.
agreed. And
Um,
we talked about domestication. Remember, like brother of another mother, domestication of
that's right.
another entire species. Yeah, there's all kinds of
Yes.
layers here. Yeah, yeah,
So Ubalubi writes, pigment progenitor cells. In figure 2D, the melanophore resembles DC's max dendritic cells. I wonder
yeah.
if they have a much more rapid environmental responsiveness compared to human melanocytes. Good question. Yeah.
Great question. Agree. And if you have some free time staring at electron micrographs of scales, iridophores, and all of pigment biology, just absolutely fascinating to look at under the microscope. And
And a follow up, could we study fish
yeah,
melanophores and their quick response gene
yeah.
expression and relate that to how human immune cells rapidly react when they recognize a pathogen?
Yeah, great question. I love this. So, I'm now thinking about things like cuttlefish and squid, the melanophores. So the creatures, that can camouflage themselves to avoid predators, octopus and cuttlefish, so forth. And so
Mm.
they're right. So in that case, and I'll concede that my, mm, sort of level of understanding here is pretty limited, but you sort of pre-deploy the pigments and the proteins, and then you move them. So you've already, right, in order to respond that quickly, um, at least for the sort of obvious charismatic traits, the ability to sort of deploy something that exists. But I think the question is exactly right, because there's going to be all sort of different levels of timing and certainly genetic underpinnings for how, like, at different time scales this plays out. And I would say we're still in our infancy of understanding that. And so drawing that bridge or that connection here that Ubaluba is making, I think is exactly like that's it, right? So thinking about, and this is basic curiosity driven science. How is it that a squid changing color relates to how our immune cells react when in the presence of a pathogen? How much of that is biologically written in things that are pre-deployed that you regulate, get into the game over a time course, or that really takes a lot of time, and then you have sort of other forces that are dictating what survives or what has a fitness advantage? Absolutely great question.
So Les wants to know, if are multiple gene copies useful to the virus, since it makes the virus persist even if a copy gets damaged?
Yeah, I think very possible, right? So, and maybe hinting at it even from this example, where there's, depending on the individual you're looking at, there might be 10 copies. The trick here is really kind of gene turnover. So as it's propagating, what shows up. So if the propagation is happening with something that's sort of genetically identical and somewhat, let's say, wounded. So let's say there's a mutation in the envelope gene, and if the rest of the machinery is good, it's active, endogenized, but the envelope copy is broken, then each time it copies, we'll still have a broken envelope, right? And so it just, again, if there's complementarity from another foamy virus that happened to show up exogenously, integrate and persist, then I think you can start thinking about exactly they could sort of potentially work together. But getting that regulation right, sort of now it's lightning striking twice, all of these things, very possible, probably rare, but yeah. And how much of that underlies the ability of viruses to propagate or to regulate things, where it's not just sort of one actor, but it sort of, approaches the complexity of real life, which is what you're describing here, Les.
Okay. I think we should do some picks, Mel's. We're running on a long time here.
Oh, geez. Yeah. This is the holiday. I'm already in the holiday mood of just sort of, settling in by the fire and letting it roll. Well, I couldn't resist, Vincent. So I've got two picks of the week. Holiday edition here. So the first one, my first pick of the week, Some Like It Hot. So this is another preprint that just dropped, I think, 2 days ago. That caught my eye. This is a geothermal, so more field work, collecting now microbes, a geothermal vent, and finding an amoeba species that sets a new upper temperature limit for eukaryotes. So we have the link here on the bioarchive. They isolated an amoeba that they name Incendia amoeba, cascadensis, divides at 63C. That's about 145 on the Fahrenheit scale. A new record for the upper temperature limit. And under, when they brought it into the lab, some beautiful microscopy, by the way, of this, and videos of this amoeba moving around at high temp, and they could see it, excuse me, using live cell imaging, try to divide up to 64 degrees C. So a real extreme, sighting of biodiversity here. I couldn't help resist thinking about, like, what does that mean for the viruses of this amoeba? Can they keep up at this sort of temperature limit?
They have to.
Yeah, they have to. Is this are there fewer viruses? Is this sort of immune strategy? No answer to that, but they have assembled the genome of this amoeba, and there's some hints about gene enrichment of genes that related to protein stability, genome stability, sensing of the external environment for these sort of hot scenarios. Okay, that's one.
Do they have endogenous retroviruses now?
I don't know. I haven't had a chance. I just saw the preprint and some videos. I haven't had a chance yet to maybe this weekend dig in a little bit and look. And, there's a very high chance that at this stage they threw away those sequences. So they might have viral sequences in there, either exogenous or endogenous. But depending on the scaffolding or the genome assembly methods they use, oftentimes the viruses and the repetitive elements associated with them are the first things thrown out, just to try to get to a sort of host level assembly. And so, I don't know the answer, but I'm curious. Okay, so Vincent, I couldn't resist. I have a second pick of the week. If you'll humor me, so this is a this is a talk, a YouTube talk from another incredible scientist I just met a few weeks ago, Kara Marshall. She is just opened her lab a couple of years ago in the Baylor College of Medicine. She works on these stretch receptors, piezo receptors. But the video here is a talk she gave to, I think, her parents' retirement community. It's a history club. And so she showed up to talk a little bit about the history of science in the U.S., the history of science funding, some of the topics we were grappling with at the beginning of our podcast, talking about some of her family connections to, the medical interventions for cancer. In particular, her sister is a cancer survivor. And so she connects her own family's and life experience with the sort of basic science, the biology that underlies some of the interventions that are possible, the clinical trials that are being threatened today, and then talks about her own lab work to, this community of retirees or folks that are, by and large, not scientists. And I just thought it was a really inspiring, well done talk in the way she connects these three things and covers a lot of bases in a way that's very accessible. So anyway, it's a YouTube video link, and we'll put that up as well here. So as you're probably guessing, part of her talk on the history of science and science funding is the threats that it's under today. And I think your pick of the week, Vincent, speaks to this pretty directly.
Yeah, it's part of the nonsense that's going on
at
NIH. This is a Science article by Jocelyn Kaiser. Let me show it to you.
Good idea.
NIH shakeup to grant decision making NIH shakeup to grant decision making sparks concern over political meddling.
Yeah.
So what's going on here? So when you submit a grant to the NIH, it gets assigned to what's called the study section, a group of experts who then, three of them typically, read it and write critiques, and they give a score. And you meet with 15 to 20 other people, and you propose a score, and then the whole study section votes. And previously, these pay lines, the pay line that you had to get better than to be funded was often published by the NIH Institute where your grant was being considered. But now this practice is being ended, and they will no longer make public a threshold peer review score needed for a grant application's approval. And so right away, what this means, in my view, is that they can fund stuff that did crappy, but which they politically think should be funded, right? Because now
Yeah.
politicians are going to be in the mix of study sections and NIH funding decisions. And, they don't want to see work on vaccines, for example. So even though your vaccine grant got a 1, which is the best score you could get,
Yep.
you don't know what the pay line is. So it's just going to obfuscate the whole process. And
Yeah, that's
so,
right.
this is some institutes don't publish a pay line already, but many of them do, and we use
Yep.
it, right? Because if you get a score, and then you could look at the pay line and go, "Oh, I'm not going to be funded. I need to get work on revising it right away." Right? It really
That's
helps
right.
you to know that. And
Oh yeah.
if you called someone, let's say if there's no pay line published, you call
Yeah.
someone, say the officer who's in charge of your grant, they'll say, "Well, we're not giving out pay lines, so I can't tell you until the decision is made." And this is not how we do business anymore. So,
Yeah. This
what
one
do you
hits
think
close.
about that, Ness?
This one hits close to home, Vincent. So, um, I was mentioning this a little bit as it relates to viral, like, so not just vaccines, like or political, anything that's been sort of caught up as a political football, but also this notion of gain of function. So I'm entangled in this, as we discussed a few TWiEVOs ago, and the situation you described is exactly where I'm sitting today. So I've got grant that turned out, it scored really well. It scored a 1, which still feels like I don't know how I pulled that off. It's the first time in my career
That's
that a
amazing.
grant got a
That means
perfect score.
everybody in the section voted 1 on Nels's proposal. That's amazing.
Yeah. And so I'm still, like pinching myself that happened. But as I then engage with the program officer, and by the way, which I couldn't do for more than a month because the government was shut down. But then now that it's sort of perked back up, at least until the end of January, I had one quick exchange, and he said exactly what you did, which is, well, I can't tell you, like, usually this score, a score, like, you can't do, I've done everything I can here, would be very competitive, but I can't tell you what's going to happen because of these political shenanigans, flipping the light switches on and off, deciding, that because we don't like Tony Fauci or we think he did something untoward, that we're going to put a shroud on this. The entire institutes might stop considering infectious microbes. This is all floating up in the air right now. And so my own research program has all of this uncertainty, whether we can have the federal support that we would usually expect to be there when you convince your peers that this work is not only worthwhile, but it's the best one that they've seen for a stretch. And so it is absolutely devastating. And it's not just me. I mean, I think I'm doing okay, but when I look around at my peers, and in particular, the younger generation of scientists coming up and the stress and pressure that's being put on them, I just saw on social media someone say, "Hey, did anyone have their grants terminated 2 days before Thanksgiving because of virology, concerns about gain of function virology?" This is someone who's doing some of the most creative, durable, robust work on influenza virus, just having the rug pulled out from under his research program, meaning that the people working on this as they go into the holiday don't know how long can I keep going. It is the AVID grants, these program grants, entire laboratories that are depending on this to drive their research, totally thrown into chaos and worse. And so, yeah, when I think you pointed out that pay lines haven't, some institutes take them more seriously than others, but it's a framework or a guidepost with some signal that you can kind of make plans for the size of your research program, the kinds of experiments or projects you can do, the people that you can hire and train to invite into science. This is all absolutely scattered to the wind. And then, go ahead. Yeah.
Go ahead. You do. You go.
Well, I was just going to say, and then we're at when we think about NIH, there's now the heads of the institutes, right? These are all open positions because these really skilled scientists who competed and were chosen through competitive processes for their science, for their leadership in science, people who were heads of NIGMS, NIAID, all of the, into the institutes, gone, rehiring them, and all of the signals are these are going to be political appointees. There's already
Hmm.
cases of this. Buddies of, JD Vance are now running the NIEHS. And so when you take that information, political appointees running these things, that article that you just shared with us information, there's a new policy that NIH is coming out with, like, oh, well, we'll just do a top down and decide. This is just absolutely throwing the whole system out the window, potentially. Really, really, I think we just, everyone deserves better leaders. People who actually have the experience and who care about this, who can help nurture and grow science ahead, not these shenanigans and,
Well, the
whatever.
problem is, Nels, that they are ideologues. They
Yeah.
know, they think that science should be done a certain way, and that's
Yeah.
wrong. Science should be done anyway. Any way you can get it done, you should do it. You don't have to say we
Yeah.
can't do this kind of science. We can't have diversity. This is all BS. Okay, and the
Yeah.
sooner we get through this, the better. Now, your 1.0 in any other time would have been funded because it's the best possible score. And it just has to get past the second level, which would be
Right.
council, and they would definitely fund it, right?
Yeah.
Unless there are 500 other labs doing exactly the same thing in the U.S., which I'm assuming there probably isn't. But now they don't want to make a pay line. They want you to decide based on the priority of the institute. So NIAID, what's its priority? Your career stage, your existing funding, and even where you are located. Because the reality is, a great part of NIH grant money goes to the East or the West Coast, right?
Indeed. Yep.
That's where the big cities are for the most part. And they don't
Yep.
like that, right? They want to spread it
Right.
out. Whereas I think it should be meritorious. You write a great grant proposal, fund
I'm
it,
with
right?
you. Yes. No, and what you're describing is major league science, right? So let's do this analogy with Major League Baseball. There's the New York Yankees, the LA Dodgers, the East Coast, West Coast sort of giants, but I'm a Minnesota Twins fan. And when they won the World Series in 87 and 91, like those are inspiring moments where the small crew comes up with something great. So we spread it all across the country, take all comers and say, let's just play and see how it turns out. And that's like, I can't think of anything more American than that in terms of the approach to science that we've been doing for
Yes,
decades. And so
sure.
now it's this pretend stuff, right? It's like, okay, imagine you show up at the ballpark and they say, oh, yeah, those really great players from this other country? Yeah, we're not inviting them to the game because why? That's not American. We want the best people to show up and take a swing for the fences. Like that's what we do. It's like that's why you show up as a fan, is you want to be dazzled, you want to be inspired. And so when you say like, well, you're not welcome, or I don't want you here, I can't, like, it's the game just lost some of that inspiration. Then imagine that they say, okay, you know what, we're going to turn off half the lights tonight, and if you're on the outfield, you don't get to wear a glove. It's like, well, hold on, this isn't the game I showed. Like, we can pretend that we're playing baseball, but you are messing with the sport. And this is now minor league. It's not even minor league baseball. This is not the major leagues of science. This is not even minor leagues of science. It's not science anymore. It's politics. It's messing around and grinding axes and all of this. And to me, that's the thing that's really the most, disheartening, is giving up on a game. And the kids, imagine that kid who grew up in junior high. He was a second baseman. He or she played shortstop, softball player, whatever it is. Small town, rose to the top, got to college. And let's say she was won NCAA championships in softball, one of the best pitchers or whatever it is. Shows up for the next level, and everyone's like, "Yeah, no, we're just going to do this kind of weird stuff where we're messing around with you." No, come on. This we For the people from generations past who put it all out there to make this even possible, to the generations ahead, we owe it to them to do our part in this process. And so it's going too far. It's the trajectory continues here. It's only getting worse. And so how do we find our voices? How do we stand up to this? How do we share this with others? How do we reclaim democracy, whether it relates to science, whether it relates to education, all of the above? And that's not to say that the old system is perfect. Look at Major League Baseball. There's cheating scandals and pine tar bats. There's always cheating, but you don't throw out the whole game. You work to do better, not work to do worse. And it really feels like we're working. There's a lot of work happening right now in this country to do worse.
605 has it right. Academics is the major leagues of the mind.
Ooh, I like that. Very
Isn't that good?
nice. Yeah.
And then one last comment from EPMG1E. This is because people do not have to face the consequences for their decision. Once a few children of politicians get a hit from preventable disease, their opinion may change. I don't think it will, because the one thing that makes people vote is the economy, and that's it. Nothing else. Nobody cares about NIH. Nobody cares about vaccines. It's the economy. Trump was elected because he said the economy was poor when it wasn't, but people believed them. And he said, I'll make it better. And the midterms in 2025, the economy is crap, and they're blaming him. That's all that matters, and everything follows. Okay?
Yeah.
I've learned this in the last couple of weeks, sadly.
No, I hear you, and honestly, and there's sort of counter evidence to the comment, right? So one of the people who has outsized power right now as it relates to scientific research, NIH, NSF, et cetera, is Russell Vogt in the office of the budget office. And, and to that, like, let's connect this down to the family level, to the personal level. The medical interventions that his daughter has been involved in, and I'm chronic disease, I'm forgetting the details now, but those treatments would not have been possible without major league science, major leagues of the mind in the U.S. And so even with that impact, and I don't know, I mean, a handful of these political, whatever, the damaged personalities, the just deep pathology, logic goes out the window. So you would think that someone with that outsized power, who has had family members depending on this very system, would have that view of, oh yeah, maybe I'm not going to just throw it into the wood chipper. I'm going to at the very least not do anything, right? Versus make it worse. Would have that view, and that's not the case with these folks. And so
his
logic is out the window.
His ideology is more important. He wants the U.S. to be a white, Christian, far-right nation run only by men. That's what all these people do, okay? And sorry, that's the fact. And it doesn't matter if we don't have any good health care anymore. We don't have any good research. All that matters is that vision of nonsense. And I don't know how we got here, but it's all about the economy. And if it stays bad, we'll flip the Congress next year. If it gets better, we're screwed.
Yeah. 2026 feels like a big turning point here for many reasons, and not just in American science. I'm thinking about these characters with outsized power in places like the Department of Defense, Department of Education, up and down the list.
Nels, these people in Congress say the military cannot do something illegal, and they're getting excoriated for telling the truth. This is crazy. Oh my God.
Pick the news headline, and it's there's some patterns here that have more than just emerged that are in play.
I saw a graphic yesterday. Countries in the world are asked to ban torture, right?
Yeah.
Every country is against torture except the U.S. and Russia, pretty much.
Yeah.
Isn't that disgusting? Nobody should ever be subjected to torture, no matter what, okay?
Yeah.
We do it all the time. Russia does it. I'm sure a lot of countries do it without saying they do. But it's just not what we are, right?
Well, yeah, I mean, that's complicated. In some sense, it is what we are, but that, aspiring to be better, so that and this is part of the asymmetry of, I think, how things play out these days, is people say, oh, well, it's more, or this is the Trump argument, it's more honest or being frank. Like, all this stuff has happened, we're just surfacing it somehow, or being more open with it. And no, you know what? Forget that. So it has, do things need to be reformed? Do we need to do better? Should we aspire to this? Absolutely. Have we gotten it right before? Has torture gone on in the U.S. since the beginning? Yes. Has slavery gone on in the U.S. since the beginning? Yes. Have all of these issues. But the thing that's changed is this idea that we want to work towards a more perfect union, as it's been phrased in other sort of political moments. And it's letting go of that impulse to try to do better, to make more opportunity. Like that to me really feels like it starts to get to the heart of this minute. But
All right.
anyway, here we
Here's what
are.
matters. Here's what matters, Nels. In Helsinki, it's -4 degrees Celsius.
There we
Hello.
go.
Welcome.
Good to see
Let's
you.
wrap it up because we've
let's get
kept
into the
people here
sauna.
long enough.
Indeed, agreed.
All right, that's TWiEVO 120. You can find show notes at microbe.tv/TWiEVO. If you have any questions or comments, TWiEVO at microbe.tv. And if you enjoy these programs, we'd like your support. You can go to microbe.tv/contribute. Nels Eldey is at cellvolution.org and over on Blue Sky, L. Earlybird. Thanks, Nels.
Thank you, Vincent. Great to be together. Happy holidays to all those in the U.S. and happy holidays across all points on the map. A foamy virus that makes you sparkle. That was really fun, Vincent.
Sparkle Pony is great, Nels. It's in the greatest tradition of cool gene names. What is one of my favorite? What's Hedgehog? And then there's a variant.
Sonic Hedgehog.
Sonic Hedgehog. Oh, it's great. Love it.
Yep.
I'm Vincent Racaniello. You can find me at microbe.tv. Music you hear on TWiEVO is by Trampled by Turtles. You can find them at trampledbyturtles.com. Been listening to This Week in Evolution, the podcast of the biology behind what makes us tick. I forgot to thank the moderators.
Yes, thank you.
Barb Mac UK and Tom, thank you so much. See you next month. Until then,
next time.
be curious. Bye-bye.